Good morning, and thanks for joining us this morning for Winship Grand Rounds. If you're an Emory University or healthcare employee and would like to receive CME credit for attending today, the login information can be found in the chat feature at the bottom of your screen. If you have any issues during the webinar or CME login, please send Julie Hawkins, our fantastic coordinator for this Grand Rounds, an email or drop a note via the chat feature. This morning, I'm excited to welcome our own Dr. Doug Graham. Dr. Graham is Professor of Pediatrics and the William G. Woods Chair and Chief of the Aflac Cancer and Blood Disorders Center at Children's Healthcare of Atlanta. He's also the Division Chief for Pediatric Hematology, Oncology, and BMT at the Emory University School of Medicine. Dr. Graham is a Tar Heel. He received his medical degree as well as PhD in microbiology and immunology from the University of North Carolina at Chapel Hill. He completed his residency in pediatrics and a fellowship in Peds Hemonc at the University of Colorado at Denver. So certainly a fantastic pedigree. He comes to us from at Aflac from having previously served as the Professor of Pediatrics and Immunology at the University of Colorado with a clinical practice at Children's Hospital there. He led the biology and treatment of childhood cancer research emphasis area for that hospital and oversaw directly all basic science, translational, and clinical oncology research for the hospital. Dr. Graham is an NIH-funded investigator with an active laboratory focusing on developing novel therapeutics for pediatric cancer, and he's recently validated and continues to grow the world of MerTK as a novel cancer agent in leukemia, melanoma, non-small cell lung cancer, and glioblastoma. He's a member of the Children's Oncology Group, COG Acute Myeloid Leukemia Disease Committee, and the NIH Molecular and Cellular Hematology Study Section, as well as memberships with the American Society of Pediatric Hemonc, ASH, ASCO, AACR, and a variety of other groups. So Doug, I want to welcome you here this morning. I want to thank you for giving Grand Rounds and certainly excited about your presentation. For those of you who are here with us, please enter questions in the chat throughout Dr. Graham's talk. We'd be happy to and be excited to answer those questions with him after the presentation is complete. Doug, take it away.
Yeah, thanks so much, Donald, and thank you for the invitation to speak today. I'll start out just with my disclosure slide. Part of what I had to do and had to learn, navigate the process of how do you translate an idea into a clinical practice. And the learning process included the realization that I had to form a spin-off startup company to enable that to happen. So the company is called Merix, and I'm a founder and have ownership in this company. So let me start out by giving you the sequence of my life in one slide, at least with relation to my scholarly work. I was a medical student at the University of North Carolina starting in 1987. And then in 1989, I decided to take 1 year off of medical school, and during that year, do a 1-year research project. During that year, as the way luck happens sometimes, I stumbled across a discovery in the project that I was working on, where a little dot on a screen, on a film, showed up an antiphosphatyrosine screen in which a new segment of a gene was detected. And so that then turned it from a 1-year project into a 5-year PhD thesis. And that started the journey with gene discovery, starting in 1989 and continuing to 1984. I then spent a number of years in University of Colorado, launching my lab, completing postdoctoral work and launching my lab after that in preclinical target validation of this new gene, which we called MerTK. The next phase began really a phase of partnering with medicinal chemistry experts in drug development, which took about another 8 years, and then starting in 2018, the launch of a phase 1 trial. And so I'm going to walk through the different stages of this cycle, going from a gene discovery into the current transition into phase 1 trials. So this was a paper published in 1994, and this was part of my PhD thesis work. And this is the cloning of the human MER gene. And so you can see on the left-hand side the amino acid sequence, and every time I look at this, I think this represents about 2 years of my life, pouring sequencing gels regularly, multiple times a week, trying to get the full sequence of the MER gene. And then realizing that in today's world, this is actually done in a matter of hours. So it's really incredible as you look at the transition in technology. When the gene was sequenced and the amino acid structure is given, we found it was very similar to another gene that had just been identified also at the University of North Carolina, and that gene is called AXL. So literally in a lab right down the hall, a sister gene was identified. And this represented 2 of 3 proteins that are in a family, and the family is called TAM, tyro-3-axle MER, and this is the axle and the MER, 2/3 of the family right here. This family is characterized by having 4 different extracellular motifs, 2 Ig and 2 fibronectin type motifs, a transmembrane region, and an intracellular kinase region. And also very early on in the studies, as I was characterizing this gene, I found that interestingly, it was expressed in many different hematopoietic cell lines, in T-cell leukemia lines, in the B-cell leukemia lines, and in AML lines. And so this was PCR. There were no antibodies, obviously, at the time, so I was looking at expression by PCR. But I found very surprisingly that there was no expression in non-leukemic T cells or B cells. And so looking at some T cell clones that were really non-cancer lines, there was no expression by PCR. So I found that intriguing early on, and then as I looked outside of the hematopoietic lineages, I also found that lung cancer lines had very high expression of this transcript as well. And so this kind of foreshadowed my future in looking at where may be an opportunity to examine the potential impact of this new gene clinically. And today, many years later, these are where the trials are focused. They're focused on looking at targeting this gene, this protein in leukemia, ALL, and AML, and in lung cancer. So there, in addition to the abnormal expression that was found in cancer cell lines, and in some cases, I was able to show that it was ectopic expression, so there really shouldn't be there in the leukemia lines because the normal counterparts didn't have it. In other cases, it was just an upregulation, so there was some expression in some epithelial tissues, and other types of cancers turned it on. But I also found that MER is normally expressed in a number of different normal cells throughout the body. So in macrophages, in a number of epithelial tissues, and in reproductive tissues. And this is how the name MER was derived. Physiologically, within macrophages, it's important in clearance of apoptotic cells. And so you can see in this schematic that that there are apoptotic debris around, and the MER receptor through the ligand, and the ligands are either Gas6 or protein S, is able to engulf the apoptotic debris. So it's involved in clearance of apoptotic cells. MER is also important in myeloid-derived suppressor cells, and it creates an immunosuppressive environment when it's activated. And it's important in platelets, allowing platelets to aggregate. And then aberrantly expressed not only in lung cancer and leukemia, but really in about a dozen different types of cancers. And we focused on lung cancer, leukemia, as well as glioblastoma and melanoma. But this picture in the lower left-hand corner is one that I love, as it shows the role of MER on macrophages in clearing the apoptotic cells. So this is showing a picture of a macrophage, surface of a macrophage. And in a cell line, a cell that has MER present, apoptotic thymocytes are actually being engulfed by the macrophage. And you can see this through the arrows here, that the macrophage, the apoptotic thymocytes are being engulfed within the macrophage. However, if the macrophage cell lacks MER, the apoptotic thymocytes can bind just fine, but they're not able to be engulfed. And so in this picture, that you can see that the apoptotic thymocytes are just studded on top of the macrophage, but the cell cannot engulf them. So we also spent a lot of time in my lab and in other labs after the discovery of MER, understanding signaling pathways that are downstream of MER, and not only looking at MER, but also the sister proteins AXL and TYRO3, and finding out a really complex array of signaling that occurs when these three proteins are activated. So downstream of the TAM receptors is ERK and P38, mTOR, AKT and mTOR, all proteins that are important in cell growth and proliferation. In addition, these proteins are important in apoptosis, in invasion and migration, as well as in oncogenic transcription and EMT. Finally, MER is important in blocking tumor growth through blocking a tumor suppressor protein, WWOX. So I'll move on now to the further preclinical target validation. After the initial characterization of MER, would this be a good protein to try to target therapeutically? And really, it branched into two different areas. And the first area is the expression within the cancer cell itself. So we found that MER was overexpressed, as I said, in a number of different tumor cells, and that these pro-survival signaling pathways were a dominant phenotype when MER was activated. So the hypothesis then was that if we could have inhibition of MER, knock down MER genetically, for example, that we could reverse the survival signaling pathways, and we could also reverse chemo resistance. In particular, the hypothesis is that we could increase the efficacy of other agents, including targeted agents or chemotherapy, when we added in a MER inhibitor. A second point is that MER is also important in the tumor microenvironment. I mentioned the role that it has in macrophages, and it's important in dendritic cells as well and other cells within the tumor microenvironment. And the role of MER normally is to suppress tumor immunity. So the hypothesis was that if you inhibited MER, you would activate the tumor microenvironment to have some anti-tumor immunity that would occur. So I'm going to first start out in the first arm of this, the MER overexpression in the cancer cells. So if we look within AML, for example, AML occurs arises from the bone marrow at different stages of myeloid maturation. In the counterpart cells, the normal cells that are in the bone marrow, the myeloblast, the promonocyte, and the monocyte, we eventually were able to develop MER antibodies, and by flow cytometry, we could show that there was really little to no MER expression at any stage of myeloid maturation in normal cells within the bone marrow. However, the counterpart cells, the leukemia cells that arose from the bone marrow, had high levels of MER expression. And so this was our early study looking at a small subset of pediatric AML patients and adult AML patients. And we found early on that somewhere around 70 to 80% of all pediatric patients with AML had high levels of MER expression. And within adult AML, about 90% of them. So turned on quite regularly within AML. And we validated this in much larger data sets subsequently. So two cell lines that had high levels of MER expression, the Kasumi1 and the NOVA1 cell line, we looked at signaling in response to addition of the ligand Gas6. And so you can see that in the Kasumi line and the NOMA line that AKT and CREB and STAT6 are elevated. And on the right, the Western blots of the different proteins, and when the ligand Gas6 is added, CREB, AKT, phosphostat6, phospho-ERK, phospho-P38. And this gave us this schematic with that within AML. MER was important for activation of ERK and P38 and AKT and STAT6, and this impacted cell proliferation and survival. So in a functional assay where we had the wild type AML cell line or a knockdown, two different knockdowns of MER, knockdown 1 or knockdown 4, or an SH control, what we're able to show is that in response to the stress of serum starvation, where blue is the live cells and green is the cells that are undergoing early apoptosis, and this upper right-hand red quadrant is later apoptosis, that there's some degree of apoptosis that occurs in wild type and SH control cells, but there's much more apoptosis that occurs when we knock down MER in the face of serum starvation. And this is quantitated below, so that relative to the apoptosis in the control cells, which is about 15%, we were getting apoptosis levels of anywhere from 30 to about 60% when MER was inhibited. And finally, if we looked at colony-forming potential in soft agar or methylcellulose assays, we found a similar phenomenon that MER was important in regulating the growth and the ability of the cells to proliferate in this assay. So we can see that the many colonies were formed with the wild type or the SH control with the Casumio #1 cell lines. But in the knockdown cells, there was very limited capability of the cell line to make colonies. And this is also quantitated down below in terms of the quality number being significantly reduced when MER was knocked down. So finally, we took our #1 cell line, which had the MER knockdown, the SH-MER1 and the SH-MER4, and we injected into sublethally irradiated mice 5 million cells, and we looked for survival. How would this impact survival in this in vivo study where MER was genetically turned off? Relative to the wild type and the SH control, where the mice died on an average of 16 to 17 days, we could double survival when MER was knocked down. So we didn't cure the mice, but we had a significant enhancement in survival. We did similar studies in other cancer models. So this is in AML. We did similar studies in ALL, in melanoma, in glioblastoma, and in lung cancer using cancer models where we showed animal models, which we showed that we had a significant decrease in tumor size and tumor growth with MER inhibition. And this allowed us to think about, with this proof of concept, that we should move into a drug development stage. And this was a new area for me to transition from someone trained in basic science and in immunology to think about, now, how do we transition at least part of the lab into this drug development phase? And in order to do that, we had to reach out and find partnerships, particularly people who had expertise in medicinal chemistry. So I went back to my roots as a Tar Heel, as Donald explained, and I found two fantastic collaborators at the University of North Carolina. Actually, my former mentor, Shelley Yarp, who was the director of the Cancer Center there, as well as Stephen Fry, who was an outstanding medicinal chemist who had just been recruited from GlaxoSmithKline to the University of North Carolina. And the three of us went in and jointly applied for a grant through the NCI, the NEXT grant, which gave us about $5 million to be able to develop the first-in-class MER inhibitor. So after many months of trial and error in continually improving the drugs that were being generated in Dr. Fry's lab, we were able to come up with 3 clinical candidates. And this was after screening about 1,300 compounds. And every week or so, our lab looking at and determining which were the best compounds of the lot that was sent to us, and then having Dr. Fry's team continually optimizing it until we came up with MRX 2843. This is a drug that's very highly orally bioavailable, had a half-life in mice of a little over 4 hours, very potent in enzymatic assays against MER in the 1 nanomolar level. But we also found that it was equally potent against another gene, FLT3. And so it's really a dual inhibitor of MER and FLT3. There is a window of efficacy against the very closely related TAM receptors. So there's 15 or 20-fold difference between hitting MER and AXL and TYRO3, and AXL is the most closely related protein to MER. And we actually did fairly extensive carnophobe profiling to show that within a range that we could dial in vitro, we could fairly selectively inhibit MER and FLT3. So we wanted to keep the FLT3 activity, particularly because of the the validation that had been previously been shown scientifically and clinically that FLT3 is a terrific AML target. So initially some of the team said, hey, maybe we need to dial out the activity. And I said, no, let's please keep it in, because I think this is a good thing to have as we move forward in clinical trials. But it wasn't by design. Again, this was by luck that our inhibitor was a dual inhibitor for FLT3. So we studied the several different cell lines that were wild type for FLT3, so there were no activating mutations that were present, and several cell lines that had FLT3 activating mutations. And so the Kasumi line and the NOMO line were two of our model lines that we thought more of the signaling was being performed through MER, and we confirmed that by knockdown of MER and how that impacted signaling pathways. In the MOM14 and other lines were ones that were FLT3 ITD, and we thought that in those lines that the FLT3 was the primary driver. And we were able to show that our drug that we had, the MRX-2843, could potently inhibit MER and FLT3 in these cellular assays. And this was in the range, you could see it by the time you have 25 to 50 nanomolar of the drug, that you're having pretty good inhibition of MER and total inhibition between 100 to 150 nanomolar, and maybe even a little bit more potent against FLT3 in these cellular assays. So if we go back and we repeat the studies that I showed you earlier with the knockdown of MER, looking at in the presence of stressing the cells, how does MER perform in the MER drug perform in apoptosis assays? We found that relative to the vehicle, when we treat with 300 nanomolar to completely eliminate MER activity, that we enhance apoptosis in this cell-based assay, this flow cytometry assay, and it's quantitated down below. So in the presence of FLT3 wild-type cells, such as the Kasumi and the NOMO1, somewhere between 150 and 300 nanomolar, we have a significant increase in apoptosis that occurs, and this corresponds very nicely with when MER is totally being inhibited. And in the MOM14 and the MB411 cell lines, a little bit more potent when FLT3 is the driver, so somewhere between 25 and 50 nanomolar, we're starting to see an increase in apoptosis and a significant apoptosis at about 100 nanomolar. Similarly, again, in the cell-based assay, the colony-forming assays, with Kasumi and NOMO1, we could wipe out colony formation by 100 nanomolar, and in the FLT3-driven lines, the MOM14 and the MB411, by 25 and 50 nanomolar, we could inhibit colony formation. So we had really nice phenocopying occurring between the drug that was developed and our shRNA knockdowns of MER, and it was nice to be able to show how our data align between the drug and the genetic knockdown. We then reached out to a partner, a scientific collaborator, Dr. Jeff Tyner, who was working with Brian Drucker in Oregon and was part of the Beat AML project nationally, and was receiving a lot of patient samples at the time of diagnosis and screening for drugs that could have activity against primary AML patient samples at the time of diagnosis. And so Dr. Tyner agreed to put our MER inhibitor in his screening platform. And each line that you see here is a different patient that Dr. Tyner analyzed in this MTS-based assay. And what he was able to show is that our MER inhibitors were very potent against a particular subset of AML patients. And so that you can see that the 0.01 to 0.1, so this is 10 to 100 nanomolar of MER concentration. A number of patients were sensitive to the drug. And if you go up between the 0.1 and the 1, so between 3 to 400 nanomolar of the drug and higher, you can see that maybe about half the patients could be sensitive to the drug in that range. Well, Dr. Tyner also looked at B-cell ALL and found the same thing to be true, that if you looked at below 500 nanomolar, you're in the range of maybe about half the patients responding to the drug. CLL, which was a subset of patients we had never even looked at before, a significant portion of those patients were also sensitive to the drug. And some patients, even with CML, had some sensitivity to the drug. So we wanted then to compare with those data, how did that compare to normal cells? So if you looked at normal cells within the bone marrow, and these are CD34 positive cells, how does our drug impact the viability of those cells, and what would a potential therapeutic window be? And we found that up to a micromolar, there was really no statistical difference in decrease in cell number and colony formation in normal hematopoietic cells. But somewhere between 1 and 10 micromolar is where we saw a statistical drop-off in the viability of the cells. So we had an idea that in high concentrations of the drug that we could have some impact on the bone marrow. And that ended up being true as we looked at animal samples. And we were able to find therapeutic windows in which we could target the leukemia cells in our animal models and not impact the marrow. But at very high levels, we could start to see some impact in the marrow with some marrow suppression. This is an example of one of the leukemia studies that we did. And so we're using our drug now at a dose of 75 milligrams per kilogram. And we picked that dose because we could show in PD studies that this dose, somewhere between 50 to 75 milligrams per kilogram, could inhibit MER in the bone marrow of leukemia cells for a 24-hour period in a single-day dosing. This dose could be lowered if we split it into twice-a-day dosing, but for single-day dosing, we needed somewhere between 60 to 75 milligrams per kilogram. And what we're able to show using this assay in this leukemia line, this is a 697 ALL cell line, that we could enhance survival from 25 to 65 days. So at the time that the wild type mice, which received the vehicle control, were dying at day 25, all the mice looked totally normal in our MER treated mice. But then somewhere around 40 days after that, the mice developed active disease. And again, this aligned very nicely with what our knockdown study showed, that inhibiting MER by itself in the leukemia assays could double to triple the survival of the mice, but eventually the mice would die. So I've talked a lot about AML. I want to expand now. I mentioned that we study this in multiple cancer models. I want to talk for just a minute about lung cancer, because I showed you early on that MER was highly expressed in lung cancer cells as well. And so we looked at primary patient samples for MER expression by immunohistochemistry and AXL. And we found that some patients had very high levels of MER and or AXL expression, and others had moderate levels. And some patients had none at all. So they sort of grouped together in different buckets for having expression of MER and AXL. But if we look at the cell lines in a panel of different cell lines, either having light or dark expression, so this is just a darker expression of the blot for MER or AXL, what we found is that the vast majority of the non-small cell lung cancer cell lines had expression of MER and or had expression of AXL. And interestingly, if you look at our control that we have on the right-hand column, it's normal human bronchial epithelial cells have almost no MER expression and very little AXL expression. So this is an upregulation of MER and AXL in these cancer lines relative to the normal human bronchial epithelial cells. So we ended up looking in a study about what would happen if we treated mice with osimertinib. And we found and have published papers in quite a bit of data showing that as mice and patients become treated that are EGF receptor mutant patients with lung cancer, become treated with osimertinib, that MER and AXL can be escape pathways that occur. So as the resistance occurs to osimertinib, these pathways are called in for the cancer cells to have an escape pathway and develop resistance to osimertinib. And we showed this in an animal model. So if we have mice in which we have tumors that are present subcutaneously in the mice, and we treat the mice with osimertinib from 36 to 90 days, relative to the vehicle, you can see that the incredible upregulation of MER that occurs in this model, as MER is becoming part of the escape pathway for osimertinib. In this particular model that we looked at, this lung cancer line, AXL was not upregulated, and TYRO3 wasn't significantly either. I mentioned GAS6 was one of the ligands for MER. In addition, galectin-3 and protein S are two other ligands that can activate MER. And you can see that the galectin-3 and the protein S ligands were also upregulated in this model as the mice lung cancer cells became resistant to osimertinib. Actually, it's a human model, but in mice. And so there's a potential loop that could occur between the activation of the ligand as well as the activation of the receptor as the mice is developing resistant to the osimertinib. We then looked at the concept then, well, what would happen if we combine our MER inhibitor with the osimertinib in this H4006 xenograft model? And we chose relative to the vehicle, we chose a dose of MER that we thought would be a little subtherapeutic. So if we went to higher doses, we might get a better response. But you can see that the treatment of MER by itself in this 20 milligrams per kilogram twice a day model has a blunting of the tumor growth that occurs in this blue line. If we treat in this xenograft model either with osimertinib or with the combination of osimertinib with MRX-2843, the red or the green, you actually have a nice regression that initially occurs of the tumor, and they overlay. So you can't tell a difference in the initial treatment phase up to 57 days, whether in the response, whether you're treating with osimertinib by itself or osimertinib in combination with MRX-2843. But then at 57 days, you stop the treatment and you look at the rebound growth that occurs. And what happens, and we have replicated in subsequent experiments, is that if you get the osimertinib by itself and you stop the treatment with the osimertinib, the tumor growth recurs. So there's a rebound in tumor growth that happens. However, if the mice have been treated with both osimertinib and MRX-2843, there is no rebound in the tumor growth that occurs. And so we look at this at day 57 and 104 and the panels on the right, and we're looking at the change in tumor volume. And you can see that at day 57, if mice are treated by osimertinib by itself, you have a decrease in tumor volume. If they're treated by the MRX-2843, about half of the mice have a response and half of the mice don't have a response with MRX-2843 by itself. The combination, all the mice have a response. And then at day 104, about half the mice that got osimertinib by itself started having tumor regrowth. Interestingly, the mice that received MRX-2843 monotherapy, if you initially responded, you still had a response. But the mice that got the combination of them have this sustained decrease in tumor growth. So there really wasn't a rebound that occurred when we stopped the drugs if they received the combination therapy. And this formed the basis of the clinical trial, which I'll talk about in a few minutes, is could there be in non-small cell lung cancer, EGF mutant patients, could there be an improvement when we combine MRX-2843 with osimertinib? So I want to quickly focus on the second arm as we're looking at target validation is what happens if you inhibit MER in the tumor microenvironment. The idea is that MER, when it's activated, suppresses tumor immunity. We can flip that when we inhibit MER and stimulate anti-tumor immunity. And I'll show you some of that data in the next few slides. So if you look at macrophage phenotypes, and this is fairly simplistically shown here as M1 macrophages eliciting IL-12 or secreting IL-12 and being tumor inhibitory, and M2 macrophages predominantly secreting IL-10 and being tumor submissive. MER is actually present in both of these macrophage types, but if you inhibit MER, what we found is that there is a transition in the phenotype from having more of an M2 macrophage phenotype to more of an M1 macrophage type, so that the cells transition from primarily secreting IL-10 to primarily secreting IL-12 in the face of MER inhibition. We started out evaluating this first in this model, this orthotopic model, in which we had a mammary mouse tumor line, which was generated from a model of the polyoma middle T transgenic mouse. We took this cell line that we had made in a black 6 background, and we injected this cell line either into wild type mice, which had MER, MER knockout mice, or heterozygous mice. And what we found in this experiment is that the mammary gland tumor cells grew just fine if MER was present. And so in the MER++ mice, the tumor cells grew, and actually the mice died fairly quickly, in 40 or so days. However, in the MER knockout mice, the cells actually never really grew at all. And in fact, you had significant survival of the mice, up to about 80% survival of the mice, in which the tumors never grew. And the heterozygous mice were somewhere in between. If we then look at the RNA level, what's happening in the cells, and so this is the polyoma middle T, and this is a melanoma line, a B16 melanoma line. And we're looking at IL-10 and IL-12 levels 4 days after inoculation. What we found is that in the presence of MER, the IL-10 was the predominant transcript that we found, but in the absence of MER in the knockout mice, that IL-12 was the predominant transcript. And then if we looked at serum levels in the mice 4 days after inoculation with these breast cancer cells or the melanoma cells, that IL-12 was the predominant interleukin in the MER knockout mice, but that IL-6 was the predominant one that we found in serum levels in the knockout mice. IL-6 and IL-12. So these pro-inflammatory cytokines were detected in the serum level in the knockout mice, suggesting that they had enhanced inflammatory states. So the other thing that we wanted to look at is what happens to the interplay between MER inhibition and checkpoint inhibitors. And our lab is continuing to study this further right now. But if we look at MER knockout mice at CD11 positive cells, and now looking at these cells that express PD-L1 or PD-L2, we can see that compared to wild type cells, that in the presence of leukemia, that the wild type cells have much higher levels of PD-L1 and PD-L2 than the knockout. So in the knockout mice, in the CD11 B cells, much lower level of the PD-L1 and PD-L2 expression. Similarly, a much lower levels of PD-1 in the T cells in these knockout mice. And it's true in the spleen and in the bone marrow. And our drug actually showed the same thing. When we treat with drug and we look at CD11 B cells, the expression of PD-L1 and PD-L2 went significantly down in the spleen and in the bone marrow. So that when we now evaluate the fact that CD11 B cells have a lot of MER present in the normal state, and the T cells, the CD8 T cells have a lot of PD-1 that's present, this allows tumor growth to occur. But when we inhibit MER, we're actually decreasing PD-L1 in the CD11 B cells and decreasing PD-1 in the T cells, and this is having an anti-tumor effect from the activation of this pro-inflammatory state in the tumor microenvironment. So one additional experiment, which lined up with the experiment that I showed you in the mammary gland tumor, is that if we take a murine leukemia cell line, this is an ARF-null P185 positive BLL cell line, and we inject this cell line, and this is a cell line that doesn't express MER, so this is expression of MER in a macrophage line and then this leukemia line. So the leukemia line itself doesn't express MER. But if we take this murine leukemia cell line and we inject this cell line into wild type cells, all the mice die very quickly of leukemia. However, if we take the same leukemia cell line and we inject it into MER knockout mice, in which we have activated the bone marrow microenvironment, we do not get progression of the leukemia. And actually the mice survive just fine, even with the injection of the leukemia cells. We're able to show this with our drug as well, that if we treated with really a very low level of dose of the drug, we wouldn't see an impact. But as we treated with higher levels of drug, where we're not targeting the leukemia cells itself because they don't express MR, but we're targeting the microenvironment, we could enhance survival of the mice just by modulation of the bone marrow microenvironment. So these data collectively in leukemia and in lung cancer led us to figure out, how do we take the next step? How do we take our drug that we have validated in our preclinical studies and transition this to see if we can actually impact patient care? And so we've launched a series of clinical trials to be able to address this question in patients. And this is where we hand the baton. So our lab was involved in helping getting the drug ready and prepared and providing the rationale. And Donald, I apologize, I'm realizing I missed the D on your name here, but we handed over the drug then to our partners at Winship and at the University of North Carolina, Dr. Harvey and Dr. Dees, who've just done a phenomenal job in doing the first in human clinical trial with the drug that we've developed. The company that I mentioned, which is really a virtual company, a startup company, partnered with another pharmaceutical company to help pay for this first in human trial, Beta Pharmaceuticals. And the first patient was dosed in June the 6th of 2018. So this was an exciting point in my career to be able to think that, 25 plus years after the discovery of the gene, we were able to have an inhibitor that was going into patients. And I'll tell you just quickly a couple of slides, and I'm not going to steal the thunder of Dr. Harvey, who will give a more extensive explanation of this trial as it wraps up, but that there were 9 dose levels that were planned for this trial, and Dr. Harvey and his team are on dose level number 7, 180 milligrams, and the trial is still ongoing at that dose level. The one thing I will say, though, which is encouraging, is that if you look back at the data that I showed you from Jeff Tyner, in which I showed you that somewhere between 10 and 100 nanomolars, that there were some patients that were starting to respond at drug concentrations in that level. And then if we went above, somewhere between 100 and a micromolar level, that more patients would respond. And actually this red line needs to be bumped up above the 1 level, because we're hitting serum levels now above the 1 micromolar levels at the dose level that we're hitting. So we would expect that a number of patients, if it were a leukemia trial, that we would be in a range in which we could be having some therapeutic efficacy. The other thing that I'll just quickly show you in terms of a biomarker from this trial is that as we would have expected, a number of patients who are getting this drug are having an increase in serum IL-6 levels. And I showed you that this was also true in our murine models, but at patients at the 80, the 120, and the 180 milligram, if you look at the pre-dose and then at day 1 and then day 15, we can see an enhancement in serum IL-6 level in multiple different patients which receive the drug. So this is exciting to think that we're at a range that we would anticipate could be therapeutic in trials, and a range in which we're having this biomarker detection as increasing. So I want to move on to a second trial, which is already launched as well. And this is in collaboration with the Emory lung Spore. So Ram and Taufik are the clinical partners in this trial. Our drug is one of the 3 projects that's being tested in the lung Spore. So we're the second project out of 3 in the lung Spore, but we have a clinical trial that's ongoing as part of the lung Spore. And this is, as Taufik and Ram have helped design and oversee this trial. This is a dose escalation in which the osimertinib level is fixed at 80 milligrams, and we're going to be, for the first time, combining it with MRX-2843, either at a dose of 80, 100, or 120, and having dose escalation of MRX-2843 until we hit the recommended phase 2 dose. And then once we find out our recommended phase 2 dose, there will be expansion cohorts in this dose expansion phase in which we will treat 20 osimertinib naive and 20 osimertinib resistant patients. So we're really excited. The first patient has already been treated on this trial, and this trial is also going to be open at the University of North Carolina. It's not quite open there yet, but it will be open there soon. And we're excited about seeing how this trial is going to evolve as well. And then finally, we have a third trial. And this trial has been funded by 2 cancer foundations, Swim Across America and the Peach Bowl Legacy Fund. So our own Peach Bowl, which is held in Georgia, has given $20 million actually to help get new drugs into phase 1 clinical trials. And this is one of the trials that they're sponsoring. Dr. Mindy Pauly is the lead PI on this trial, and she is a pediatrician in our program within Aflac, and she's partnering with Bill at Emory. So this trial will open up for the first time. A trial will open up at Aflac and at Winship at the same time. And so it will be adults and adolescents down to age 12. So we're really excited, not only about the drug, but the fact that we're going to have the same trial open across the street at both centers. In addition, this trial will open up at the University of North Carolina. And so we have a pediatric leukemia doctor as well as a medical oncologist that are participating in this trial. And then encouragingly, we've had a couple of other sites that have contacted us about opening the trials at their center. So we're exploring a couple of areas in which this trial could open as well. Starting us on this trial will be 120 milligrams, but because there's funding available, there's lots of biology that's planned with this trial in particular. So we have some RNA sequencing and single cell sequencing and cytokine and signaling studies that are going to be done. And this trial is almost ready to launch as well. So we have the IND in hand. And we're just finishing up a little bit more of the paperwork, but we anticipate within the next few weeks to be able to enroll patients on this trial. And this will be relapsed and refractory ALL and AML, as well as mixed phenotype leukemia patients. So what do we do for the next steps? Where do we go forward? And I can just tell you, there are other trials that we're already thinking about. What if you have a trial, a solid tumor trial in conjunction with the checkpoint inhibitor? Or what about a true pediatric leukemia trial that would include patients below the age of 12? And so we're exploring what are funding opportunities to open those types of trials as well. And I can say that in addition to the study that I showed you with osimertinib, we've done combination studies in different types of cancers, including combination with chemotherapy. And whereas we can't cure the patients with monotherapy of MRX-2843, we even get a much better survival when we combine with chemotherapy or other targeted agents such as the osimertinib. But we've started to try to look at understanding the next steps. Why can we not fully cure the patients, which we know should be sensitive to MRX-2843? So why are we having some partial resistance, particularly in the bone marrow? So within leukemia cells, we actually can clear leukemia just fine in the periphery, but how can we clear the leukemia more effectively in the bone marrow? And so we've started to look at patients that are very resistant to the drug based on Jeff Tyner's data that I showed you, and patients that are very sensitive, and finding genes that could predict sensitivity or resistance to the drug, and started to look at pathways that might mediate resistance to the drug. And I've shown you some of them here in this schematic. But we've used that information then to look at how we can enhance combination therapies. So we have certain pathways and processes that are being targeted in the lab in combination with MRX-2843 that we've pulled out either from RNA sequence or drug screening libraries. And we're actively investigating these different types of combination therapies that could occur to be able to enhance the efficacy of the drug. And finally, I'll say we've had some really exciting progress recently looking at drug delivery. So in collaboration with Eric Greden's team and the biomedical engineering, his lab is actually here at Emory as part of the combined program between Georgia Tech and Emory. He's been able to develop a nanoparticle which has encapsulated the MRX-2843 and has really shown pretty stunning ability, particularly in the leukemia models, to have enhanced efficacy. And I don't know if it's because the drug is being more selectively taken up by the leukemia cells in the marrow. We don't quite know why. But an example is that in this really pretty resistant model of leukemia, in which the MRX-2843 isn't doing much at all by itself, we can show that when it's delivered, and this is daily delivery, we can show that delivery once a week by the nanoparticle, we get significant enhancement in survival. So this is going from changing a pretty resistant model into a somewhat and fairly sensitive model. And other models that are already sensitive, we've even shown even more impressive survival outcomes with the use of the nanoparticle. And we're exploring maybe even combination therapies within the nanoparticle. So we think that a way that we deliver the drug in particularly in leukemia models, but maybe even in solid tumor, could even enhance survival further. So I'll stop here and just say that this has been an exciting collaborative opportunity for me to partner with many different people in this sort of team science to drug development that has occurred between my lab and the laboratories at University of North Carolina. So Dr. Earp and Dr. Fry are the two main collaborators that I had in Chapel Hill in this project. Xiaodong Wang is the medicinal chemist who partnered with Stephen Fry. And the UNC team is shown in the lower right. And my lab is shown in the left here. And I'll say in the lab that really the lab has done a fantastic job of having both the basic science and the translational and being involved in drug development. And all the work that I have shown you, particularly in the last 10 or 12 years or so, has been in collaboration with Dr. Deb de Riker, who has been a partner with me in the lab and came with me from Colorado to Emory. And she and I together have supervised the projects that have been ongoing in the lab. This is a picture, our Brady Bunch picture of our lab in one of our recent lab meetings where everyone's acting a little bit crazy, but we have a fantastic team within the lab. And I want to thank everyone in the lab for being able to advance the work to get our MerTK inhibitors into patients. So Donald, I'll stop here and see if we have any questions.
Thanks, Doug. Fantastic talk, as always, and no worries. Donald is my Irish name, so that fits well. We have a question from Ned Waller. Great talk, Doug. Given the role of MerTK in platelet aggregation, have you seen any toxicology signals in preclinical or clinical models related to bleeding?
Yeah, great question. And we, through people who have been collaborators that are interested in platelet biology, we've investigated that question quite a bit with MER knockout mice and with our MER inhibitors. And what we have found is that MER is not important in sealing off a rupture of vasculature. So the platelet cells can form a monolayer just fine without having MER present. But what MER blocks really is the aggregation on top of that monolayer. And so it blocks the stable clot formation that can occur. So if you look at MER knockout mice, they actually don't have any change at all in terms of their bleeding time, but they have a decrease in thrombosis that occurs. And we've been able to show in models of pulmonary embolism and thrombosis that we can significantly decrease thrombosis and pulmonary embolism in MER knockout mice. And we can decrease thrombosis and pulmonary embolism in thrombosis in a cremaster muscle model with our drug. And so we published that as well. We've thought about at one point, would this be a good thing to advance as kind of an antiplatelet drug? But it takes so many patients to explore that right now we're sticking to the oncology indications.
Yeah, I'll hopefully the predictive models and other things would help to advance that and reduce numbers, but certainly appreciate those challenges. Another question from Ram is that, Doug, great talk. In the clinic, it's been difficult to combine small molecule TKIs with PD-1 inhibitors, and that's certainly been true with hepatotoxicity as an example. Have you seen any toxicity issues in preclinical models with a combination of the MerTK inhibitor and PD-1 antagonists?
Not yet, but we're just beginning to explore that. And so far it seems to be fairly well tolerated. And Donald, I know at some point you'll give us the clinical review of MRX 2843 in patients, but in general, the drug by itself, at least at the levels that we've evaluated so far, it is well tolerated in patients. And we haven't seen enhanced toxicity in our preclinical models when we've added in the PD-1 or the PD-L1 inhibitors yet. Well, I mean, we have to do more studies, but so far it seems promising.
Yeah, no, it's certainly the linear PK, the exposure data looks great so far. And as you say, we'll wrap the study up here in a little bit and hopefully have full results. A couple of other questions in the chat. Doug, great talk. Have you looked at the mice that acquire resistance to MerTK inhibition using some genome-oriented approach to understand the escape mechanisms? And that's from Manoj.
And absolutely, that's where we're pouring a lot of effort right now. And we have mice that we're going to be studying, as well as the human patient samples, and doing a lot of the studies, even with our future patients that are on trial, to find out, how's the gene signature being changed when we're treating with the MRX 2843 in patients. But we're just beginning to understand these resistance pathways, and what we can say is that we know that there are some things, particularly within leukemia, in which the marrow stromal cells and other cells within the marrow are helping mediate this resistance. And so we have one of the thesis projects for our graduate students right now is evaluating the fact that IL-6 could be an important way in which there is being resistance to cells within the marrow. And then if we add in IL-6 inhibitors, actually, it helps reverse some of the resistance that we're seeing in the marrow. But we're looking at other targets as well to be able to find out, can we reverse the resistance that is occurring in the tumor microenvironment?
Okay, great. Another question. Congrats, Doug, on a fantastic talk and amazing work bringing MerTK inhibitors to patients. Given the effects on the PDL axis, have there been thoughts to combine your inhibitor with PD-L1 inhibitors in non-small cell lung cancer? I think you talked about that a little bit.
Yeah, and I actually just talked to Ram about that this week. Like, when can we start that trial? So we have to get the funding to do it. So one of the things that would likely be the subset of patients that we would look at is that in EGF receptor mutated patients, there doesn't seem to be a significant response to these checkpoint inhibitors. But the ones that are not EGF receptor mutated, this could be an area which we could explore combination therapy with a MERTK inhibitor and a checkpoint inhibitor. And we have shown, even though our Spore grant is focusing on EGF receptor mutated patients, we've shown in other subsets of patients which are not EGF receptor mutated that MRX 2843 has efficacy as well. So we're actively looking at a way to be able to see if we could fund that trial too, Andrew. But thanks for the question.
Yeah. From Nikki, a fantastic talk given the role of macrophages in the CNS and neuroimmunity. Have you seen an adverse CNS signal in preclinical studies?
Not yet. So there is a study that shows, and we didn't perform this study, that shows that actually MER inhibition could be important in decreasing the ability of the leukemia to spread in preclinical models to the CNS. And so we don't know yet if that's going to be true in our patient samples. And we also are going to have to wait and see how much active CNS penetration we have in patients. We expect, based on the animal models, that we are having some CNS penetration with the drug. But that's a good question. So far, we haven't seen toxicity with the drug, CNS toxicity with the drug in preclinical models, or in patient samples, or in humans as well.
No, it's certainly been very clean in the phase 1 trial in many aspects, and CNS has not been one of the adverse events we've seen so far in the escalation or at the higher doses. Okay. I think that's it for now for questions. We'll give folks a little bit more time here, but otherwise we're nearing the end of the hour. Doug, again, great talk. I want to thank you for coming and presenting on this as a tour de force of discovery all the way to first in human testing. And we certainly look forward to hearing more from you and others about where the compound goes. So.
Thank you. And thank you, Donald, for all you're doing for the phase 1 trial, and really everyone. This has been a fantastic move for me to come to Emory and be able to have the trials launched and have the partnerships that we have with everyone at Emory. So thank you so much for all you're doing.
Thank you. Appreciate it, Doug. Take care.
Okay, bye.